Welcome to our third edition of our panel discussion, Stimulating Growth, Cultivating Solutions by Eppendorf. Our third topic today is Streamlining Biologics Scale Up. I'm Stefan Goldenbaum, I'm your moderator. Biologics have been in biotech accounted in 1995 for 15% of their entire pipeline. Nowadays, they overcome already more than 40%. This development we would like to discuss with our four guests today. Here in the studio, I would like to welcome Klaus Ferber, Head of Key Account Management at Eppendorf. Further, Winston Wong, Head of Commercials for Bioprocess in Americas. Ben Escher, Senior Product Manager for Bioproduction at Thermo Fisher, and John Shyu, Director Scientific Applications at Corning Scientific. Welcome to all of you.
Thank you.
Klaus, you came in contact during your business life with a lot of big pharma and biopharma companies developing projects with bioprocess applications. To start with, what are, from your experience, the main criteria for biopharma customers from an economical point of view?
First of all, biopharma customers are typical companies. Their business model and their philosophy for doing business is have sustainable, profitable growth. Mm-hmm. So a lot of these factors that apply for other companies from other segments do also apply for the pharma industry. So if you look into a normal setup of a profit and loss for a pharma company, typically they are generating around 16% of cost related to R&D, beside another bucket of 28-29% that is related to sales and marketing. Mm-hmm. And that, with the gross profit margin that they generate, leaves a net income of maybe up to 14%. Mm-hmm. So one of the biggest threats I could see right now for the industry is a regulation of medicine prices. When you look at the drug market as such, the volume is roughly $1 trillion annually, and that is split in, let's say, the Americas around 40%, Europe maybe 22-23%, and the remaining part is then in APAC, Australia, Japan, and also Latin America. Mm-hmm. So with influences of certain governments on the pricing for drugs in their markets, it can easily have a detrimental effect for those pharma companies. So remember, I just said the net income is around 14%. So if one of the markets is reducing or regulating pricing significantly, it will trickle down to the bottom line and reduce the net income significantly. Mm-hmm. Looking at the cost side that these pharma companies incur besides production, if I'm focusing a little bit on research and development, of course the biggest bucket is research and development. Mm-hmm. And that is one of the triggers that can be controlled by a pharma company, and therefore there is a lot of focus and a lot of pressure on those kinds of budgets. Mm-hmm.
And then what is the impact of this budget pressure towards drug development?
Typically, when you are splitting this budget, you have a couple of components. Number one is equipment and instruments, number two is consumables, number three is chemicals, and number four is resources, human resources there. And whatever application you run, whatever experiment you run, you have the choice to mix all of those four. So there can be, at the discretion of the lab manager, he has to choose where to put the money. Mm-hmm. The impact that such budget pressure has on research and development should not be under-evaluated, because at the end of the day, that experiment needs to work. If you are, for example, keeping in mind the cost that each of these pharma companies have to incur for experiments that are not reproducing the next time they do that. Mm-hmm. That is a huge factor, a huge, enormous cost factor in the overall research and development budget. Mm-hmm.
Thanks, Klaus, for giving us an overview about the economical aspects in biologics and drug development. We also would like to shed a little bit more light into other aspects for this topic. Therefore, we have as our next guest with us Winston Wong. Winston, you have 23 years of commercial experience in life science, focusing strongly on equipment, consumable, and service, especially during the past 10 years with bioprocess applications. Welcome, Winston.
Thank you, Stefan. Glad to be here.
From your experience, what about operational challenges, especially when working at process transfer from R&D to production?
Yeah, I think it's a great point that Klaus made about the economics, but clearly there are many challenges, as you mentioned, that are also with the operational. You know, when people scale up, one of the big things that we also think about is the fact that you're also increasing your financial risk, as well as the risk, for the project, to succeed or fail, because there is the economics of the amount of material that you're going to be scaling up to as you're going to that next level. So therefore, it's really important that, people take the appropriate time and consideration that is needed in their scale-up strategies to make sure that they're doing it right and that they're getting it right the first time and not having, increased risk for failure. So when you start to think about that from, kind of a high-level basis, one of the first things to think about is what is the scale that is actually, in terms of scaling up, that is actually needed? When you first ask this question, this then helps you then lead into things like first about what kind of facilities are you going to need in terms of space and footprint. Talks about then what kind of equipment that you're going to then need in order to place into that facilities in order to operate at the scale that you're going to want to produce. And then third, really then leads into the resources. And Klaus mentioned this a little bit also in terms of the people resources, because to operate at these higher scales, you're going to need additional people with the appropriate expertise who can operate those successfully. The other things that you think about from an operational challenges can then be, of course, what kind of environment are you going to need to actually set this up in? Is this going to be a clean room environment or not? Or is it also going to have to be a GMP or CGMP type of facility where you're going to have regulatory oversight? Mm-hmm. When you ask and answer these questions, this helps to address some of, once again, the operational challenges as you're scoping the entire project of scaling up. Then you can start to get into some of the details about what type of equipment are you going to use. Facilities in scale up, traditionally can use, closed systems where you're doing either sterilization or clean in place types of applications. Or you can also set up your facilities with single use, which is a very common also mix of practice in today's scale-up strategies. In addition, you also have to think about, is your upstream process going to be matched to the capability of your downstream process? So if you're capable of producing a lot of material as you're scaling up, but you forget to think about whether or not the downstream applications can also process that material, then sometimes you will have a mismatch, right? And so that's an important technical operational challenge to also take into consideration in your overall strategy. And lastly, the questions that from an operational perspective is, what is the timeline often that customers desire in order to get this next scaled up facility operational, and how would they test it along the way to ensure that it becomes successful when it needs to be ready? The last piece, is once again, the staffing. And when you then realize that you need to hire and staff these people, some of them may come with the appropriate experience, but some also might need ongoing training in order to make sure that the operations move smoothly through the use of the equipment on a day-to-day basis. And this is really where I think vendor partners, as well as other institutions, can really collaborate to ensure that that ongoing training, service, supports, and applications is really, really well met. So that overall you are, addressing that operational challenge to ensure that the people there are the ones ultimately producing the best chance of success.
Yeah. Thank you, Winston. You gave us a lot of insights into the operational challenges, also challenges involving human resources, also attached to the equipment used and also required for drug development. Where do you see technical challenges?
Yeah, so technical challenges like the economics and the operational is also a very strong area that needs very careful consideration. And that is because, when you're thinking about customers going through the challenge of thinking about how are they going to produce a scaled up facility, there are many different known approaches. And one thing, even though these approaches have, specific methodologies of calculations that you can use theoretically in your scaling up strategies, oftentimes none of them will give you a surefire guarantee that they're going to be successful. So there will always need to be some significant work and further optimization as you scale up and test those different scales up to make sure that you're conserving the same types of, quality of yield output that you ultimately want to produce reproducibly. So when you think about the technical challenges, just as an example, one place that people can typically start is something that we call a conservation of the supply side. And what I mean by that is if you can, since bioprocessing or bioreactors have many engineering design elements, if you decide to go with a scaling up strategy, one of the things you can immediately do to reduce risk is try to conserve some of those design elements as your equipment goes to different scales. So things like, for example, your height and width geometries of your vessel, making sure that specification stays as close to or identical as you go from, for example, like a 10 liter to a 100 liter or 100 liter to a 500 liter type system. Other aspects of your design elements might include the impellers or the headspace in the vessel itself, your sparging geometries and strategies, and your placement of sensors. So some of these elements that if you can conserve from the supply side will greatly reduce the potential for the variability that could be introduced as you go into different scaled up types of equipment. Then when you start to think about the actual methodologies of the approaches, once again, there are many. And, I'll mention a few here that are very commonly used throughout industry, but whether you're doing a fermentation or cell culture, any of these methodologies can generally be considered. Typically industry, what we see is that if you're doing fermentation, people start with tip speed as a very common methodology and seeing whether you can conserve tip speed as you're moving up to different scaled production. But then tip speed also can be used in conjunction with something called OTR, which is the oxygen transfer rate. Mm-hmm. There are two other very common formats, which is something called a KLA constant, and also then a power per volume constant. These two other methodologies sort of round out the tip speed as well as the OTRs, which are commonly used. What we see mostly in fermentation applications is that customers typically like to use a combination of the OTR mixed with the P power per volume, whereas in cell culture applications, tip speed really with the KLA is the most common formats of combinations that's used in the scaling up strategies. That math then really helps to set some of the key parameters around your agitation to ensure that the fluid dynamics are conserved at the various volumes that you're scaling up through. Even with all of that approach, those different approaches, there are always going to be cases when you're scaling up where you're working with a challenging, biomolecule that may not necessarily just fall into an easy scalable approach. And we see this routinely throughout industry. So in those cases, customers, even with conservation of and using the combination of these approaches, may still have to do process optimization at that higher scale. Mm-hmm. And that may be a result of just the complexity of the molecule itself or some of the factors related to the fluid dynamics, just as some examples. And I think that this is really where collaboration in the industry for scaling up strategies becomes vitally important, because when you have the scaling up strategy, it becomes very important to, once again, reach out to others who have already experience with that, as well as the vendors and manufacturers who have a tremendous amount of experience with operating their equipment with a variety of different species that are growing these biomolecules in. And so from a technical perspective, these are some of the theoreticals that scaling up challenges present. And, there are other things that then go beyond that, which some of my colleagues will comment on here that relate to, for example, then IT networking of the entire equipment throughout the facilities, as well as then data management, which in itself is also another very significant technical challenge.
Thank you, Winston. Connecting the economical challenges with the operational challenges, which brings us to our next guest, shedding a little bit more light into the automation aspects of biologics in drug development. Ben, you have 25 years in bioprocess, developing skills in visual analytics, data mining, and business intelligence. We would like to link the operational and the economical aspects now a little bit more to automation in biologics for drug development. Welcome, Ben.
Thank you very much.
Ben, with your experience, especially in automation in the field of bioprocess, how can bioprocess equipment help streamlining biologic development and scale up?
So first off, I'd like to thank everybody at Eppendorf for being here, and not only for being here, but for choosing the Thermo Scientific High Performance Sub for use with the BioFlow 720. This certainly shows a best-in-class approach to scale up, given the topic that we're talking about today. Looking from an engineering aspect, like Winston had mentioned, talking about tip speed, KLA, geometries, sensor location, all of that is standard throughout the entire high performa subfamily. One of the areas that I wanted to touch on in regards to scale up is in regards to those controls. And when we talk about controls, we need to look beyond the tip speed and KLA. We need to look at how are we having control from R&D to process development to manufacturing, where we ultimately want to go to. So if we look at something as simple as dissolved oxygen, and we look at how we can control dissolved oxygen, is my controller controlling it via agitation? Is it doing it through increased airflow? Is it doing it via O2 supplementation? And even further than that, when we look at scale up from that R&D to process development to manufacturing, look at something as simple as dissolved oxygen. Even O2 supplementation can take a different meaning depending upon the type of controller as well. So are we using rotometers? Are we using solenoids? Are we using mass flow controllers? Those can play different parts in our ultimate scale up. We see time and time again that CROs and CMOs have processes that say maintain at a set percentage, 35%, 40%. But how did you get there? How did the system actually control that? Those can play a big part in your scale-up design, and having a controller that's unified throughout that entire R&D process and manufacturing can really assist with that scale-up and ultimately reduce that time to market.
You talked about aspects in automation, also a lot about aspects of control that are required here for process setup. When talking about automation, then we also have to talk about certainly result generation and data management. What do you see here for the aspects of data management and documentation?
Well, absolutely. That's a great question. And certainly we're all driving to Pharma 4.0 and really trying to get those Pharma 4.0 capabilities of having a true paperless lab and being able to achieve analytics across your R&D, process development, manufacturing. And the best way to do that is by reducing data silos. Having the same type of controller or the same data output certainly will reduce that, and also will increase our ability to do analytics, another key factor in Pharma 4.0. Even if we look at something like, again, an example of a biomass sensor or some type of analytical tool that we incorporate at the R&D stage, but we also want to have that at the manufacturing stage. Having a control strategy or an automation platform that allows you to reuse that integration can reduce that risk and can ultimately get us to market quicker. So again, having a DCS or a distributed control system that's able to do R&D, process development, manufacturing can really reduce our time to market, reduce our economy as our economics of scale by having to be able to reutilize a lot of our data mining, data and data storage techniques that Klaus touched on in the beginning. So definitely data is a key part when looking at scale up.
And talking about all of these different kind of aspects that have to be paid attention on, operational aspects, the aspects of automation you introduced, also data management, control process. Where do you see the major pain points for pharma companies equipping their facilities?
Well, certainly as we start moving more and more to single use, Winston touched on one of the conservations of supply chain. And really, we need to understand our bioprocess containers, our BPCs. And one of the things that we need to understand about our BPCs is the film type. And you asked about a major pain point. And one of the areas of validation and certifying different types of film type can be one of those bottlenecks. But having a film type that's used from media storage to buffer storage to your bioprocess container, and then for your downstream processing can really reduce that risk and not have to revalidate different types of film types. So another area outside of those kind of engineering areas that we talk about when we look at scale up, but looking at not only scale up, but reducing overall costs like Klaus talked about, and then also really having reusing a lot of our resources or being able to reuse those validation that we've already done. So when we have different types of film type, it really puts in bottlenecks and revalidation. And then also on the supply chain side, not only looking at the film type, but understanding the vendors that are able to deliver those bioprocess containers and making sure that they have multiple sources so that you're not impacting your supply chain and your ability to make product, and reducing those lead times as well. So, working hand in hand with vendors is key to reducing that pain point in biopharma production.
Thank you, Ben, for sharing your experience. And that also addresses some of the time-saving aspects you mentioned before using single-use technologies. Right, Klaus?
Yes, correct. Yeah. It's interesting, definitely interesting to see that these kinds of corporations are functioning and the impact that could have in order to support our customers in the pharma industry by achieving their budget goals.
Thank you, Klaus. Yoshi, you have 12 years of experience in vaccine development and cell therapy. So far, we have heard a lot about automation, operational aspects, as well as economical aspects. You are coming more from an applicational point of view. Can you share some insights into applications and their challenges?
Yeah, absolutely. So my colleagues were able to talk about the importances of automation, operational, and economical aspects about bioprocess. But one of the most important things for me, from my perspective, is the notion of applications. So what do I truly mean with applications? So new technology is going to come and go. A lot of folks, a lot of manufacturers will be able to make new technologies, improve technologies to address aspects about automation, about operational economics, and how to improve the process. But one of the most important challenges that I have seen in the last decade or so is the ability to translate all these new technologies into proven processes. And what do I mean with that? So a typical vaccine production or typical biological production uses a very defined process, a very defined biological process that has been implemented maybe 10 years, 20 years into the manufacturing steps. And when you come in with a new technology and want to introduce that new technology to improve the process, a lot of times you are stuck in a process where you cannot translate the work. The biological aspects of being able to take an old process into something new just seems to be a great challenge. So that's where the applications come in. What we have noticed is if you give a R&D group, a research and development group, a technology transfer group, or even a process development group, a new product that they might not necessarily understand all the benefits of it, it is really hard to take the science and be able to translate it right away. They might have challenges in the scale-up process, something that they optimize in a smaller bioreactor, try to take it into a larger bioreactor. The proportionality of the process is not there. So that's one of the things that we always want to address on the applications perspective, is that you have to look into the biology of the product, understand the biology of what is happening with the production and how you can complement that to a new technology or the new technology that is coming in. That at any given time is going to improve the economical aspects, the operational, or even as Ben mentioned earlier, there's a lot of data that comes with it. How do you translate into something functional at the biological level?
And then can you give us here a typical example from the industry?
Sure, absolutely. So a very typical aspect as to what happens is there is what I call two different schools of how can you produce something in a biological form. What I mean with that is there's two types. You either have adherent cell lines or you have suspension cell lines. From a physics perspective, having something that is produced in a suspension, it's always going to be more beneficial than something that is adherent. An adherent platform is what we will call in the industry something that is a little bit more traditional. Historically, is the process that has been established, but it does take up a lot of space, right? Winston talked about what is the floor space that is needed, what are the economics, the impact of having such a large space. So when you're starting to bring in technology that can really truly change the total footprint of how what is required to make that, a lot of times the science just doesn't translate. Let me give you an example. If you are used to working with planar technology where your cells are growing in an adherent platform, you want to now bring it into a suspension, you have to adapt the cells into suspension. The adaptation, the steps of adapting that cell to suspension literally generates a lot of challenges. You have to deal with proportionality, you have to deal with shear stress, you have to deal with epigenetics. The epigenetics of a cell line that was making something when it was adhering, it might not necessarily make that biological product when it goes into suspension. So at the end of the day, there is a lot of hand-holding, not only from a technical aspects, but you also have to work on the science. So my best advice is we can create a lot of great technology, but you also have to create great technology that works for great scientists.
So what you are saying is that changing a certain technology very often also requires changing a lot, if not all, the processes in a laboratory that have been in use before, right?
Yeah, absolutely. So what you just said is correct. A lot of times when you bring a new technology, you are changing a existing and proven process. The process might not be the most optimized with today's conditions, but being able to translate that and be able to show the benefits of the new technology, it does take time and it does take a lot of hand-holding and optimization.
Thank you, Yoshi.
You're welcome. Absolutely.
So we have learned today a lot about biologics and drug development, especially, as we said before, from an operational point of view, from an economical point of view, as well as shedding light into the topics of automation and application. I would like to thank the panelists for their time and expertise today. Thanks, Klaus, for being here.
Welcome.
Thanks, Winston.
Welcome.
Thanks also, Ben, for sharing your expertise in automation, as well as Yoshi giving us an insight into the application aspect. Thank you all for your time and participation today. Visit our website to learn more about Bioprocess for upscaling solutions and other applications. Looking forward to welcome you for our future discussion rounds. Thank you.